All right. In this section, I want you to go through a series of steps that are involved to solve any sort of genetic problems. So first, let's look at in genetic, how do we know a trait is dominant or recessive in humans? Now, here in this picture, this is a family tree which is showing individuals that are affected by, for example, this particular family tree is showing individuals that are affected by the disease. And those that are affected, for example, are shown by the double A or the two lowercase a. And this would be for instance, notice that these are both recessive traits. So an individual who has both of these is going to be affected by the disease and will be sick. So it actually doesn't matter which genetic disorder you're looking at. I just want you to think of any kind of genetic disorders in which an individuals with a heterozygous or a homozygous allele can show either the disease or it might be the carrier. Now, the one that is yellow is of definitely the disease or the affected person. For example, when in this particular example, there is one allele that is the affected one, but there is the dominant allele, which is the uppercase, and that is the uninfected. So this individual is going to be not carrying the disease because it's a heterozygous individual. Now, if we want to know what is the genetic genotype of the parent of these individuals, then we can detect that by the pedigree. Now, from this pedigree, you can see that the two affected parents will always have an affected children. So, but since both of these children are not affected, so this indicates that the parent must have been capital A, or the dominant or the unaffected, because when you if both the parents, if this parent was, for example, heterozygous, then one of these children might have been affected. However, let's look at what happens in the second and the third generations. Notice over here again, when the alleles separate, you're showing that the third generation, all of them are again heterozygous. So this means that this person is also either another double A or the uninfected. Because we still don't know the alleles of this one, but because the child didn't have it, so it's assumed that there's one unknown allele. Now notice after a series of generation up here, you see the diseased children or the affected children, and you can see why, because the homozygous recessive is now being now expressing. And both males and females are affected with equal frequency if it's an autosomal recessive disorder. Now, many times a pedigree shows the pattern of inheritance for a particular condition. And up here in this picture, particular one, usually the squares are represented by the males, and the males are generally the squares, and the round ones are generally the females. And you can see that there's really no effect of the male and the female. Both males and females are affected with equal frequency. So we use the term carrier when we see it's a heterozygous individual which is not really affecting the disease, but they may carry it on to the next generation. Close relatives who reproduce are more likely to have affected children. Okay, in the next couple of slides, I want you to understand the 5 steps that are involved to solve any genetics problem. So for the first, the rule is that code the alleles, identify which is dominant or recessive. So for example, we said earlier about the earlobes, attached or unattached, the pea pod color, the flower color. So the one that is expressed is always the dominant alleles. So first you must identify which is the dominant and which is a recessive. Always determine the parental of the genotypes of both of them, or the given genotypes. That is, are they both homozygous or heterozygous? Next, parental or given gametes. So learn how to make a gamete. So for example, if these are, actually I'm going to take this off. We'll first pick up a characteristic. So for example, let's say rolling of the tongue is a dominant feature. So now we can call that as capital R as the dominant, and the small r could then be the recessive allele. Now, if we are talking about parental genotypes, is it capital R, or is it capital R, small r, or are they small r, small r? That would be the second step. In the third step, you must learn to separate out the gametes, which will be separate or pull this combination and break it apart. Again, if I do the same thing over here, I get one capital R, one small r. And if I do the same thing over here, I get small r and small r. So that would be the gametes. Next thing would be you make a Punnett square, and you would you have the male gametes on one side and the females on this side, and then you start putting them in boxes. So if you can get this concept, it gets a lot easier. So let's try it in humans. Being able to roll your tongue is a dominant characteristic. So if a heterozygous woman marries a man who cannot roll his tongue, what are the chances of a child of theirs won't be able to roll its tongue? So now notice we know that the dominant allele is capital R, so the recessive must be the small r. So you always pick up the same alphabet, just have upper and lower case. So we're talking about a heterozygous woman. So think about it. What would be the genetic makeup of a heterozygous woman who marries a man who cannot roll his tongue? This indicates that the man, if he cannot roll his tongue, has both recessive traits. So the first step would be the heterozygous woman would be capital R, small r, combines with a man who cannot roll his tongue. So that would be lowercase, lowercase. So this is my first step. In my second step, I am now going to code the alleles and identify which is dominant, recessive. So which I have actually done that in the past, in the previous slide. Let's go back again and do it like this. In humans, being able to roll your tongue is dominant. So that means the alleles are like this. And now you know that capital R is dominant and lowercase is recessive. That's my step one. Okay, let's go on to the next step. In this step, now I will get to identify the alleles, which are going to be capital R. Actually, it's going to be the same. I went a little ahead in the previous slides, so that's why it seems like the slides are repeated. So this is the genotype of the father. The male, and the female would be this. Okay, let's go on to the next step. Now in the next step, I have to get the gametes. So I'm going to go back and write down my mother crossing the father because he cannot roll his tongue. So now the gametes are going to be separating each one of them. So the two gametes that are formed from here would be small r, and from here it would be capital R, small r. So just remember the gametes that we just made. Because why? Because remember, in meiosis, the alleles are present in homologous chromosomes and they separate. So the alleles would be separated. Now, in the next step, you will then draw a Punnett square. A Punnett square will be making, this is a monohybrid cross because there's only one characteristic. So I will only have 4 boxes. My alleles will go on the top. So let's say I put down the mother's allele on the top. Notice I just put one separate allele. The father's allele go over here. Do you notice? One only if it's a monohybrid. Now the next thing would be to put the combo in the boxes that are shown here. And now that I've done that, I need to interpret what does it indicate. This individual will be dominant and can roll their tongue. This individual is also dominant, so there are 2 dominant. This one will not be dominant. This is recessive, and so is this one. So that means that recessive children will be 2 and 2 dominant. So that, if I look it up, it's a ratio of 1:2, or we can say 50% chance that the child will be able to roll their tongue and 50% that they would not be able to roll its tongue. I hope it made sense to you. Now, if you reread the problem again, you should be able to answer the question correctly. In humans, being able to roll the tongue is dominant. So if a heterozygous woman marries a man who cannot roll their tongue, the chances of their child won't be able to roll its tongue is 50%. So this is an example of how you would do a genetic problem in your exam or in your test. Now, let's do another one. In this one, it's a homozygous tongue roller and a non-tongue roller marry. What are the chances that the children will be able to roll its tongue? So notice homozygous tongue roller, which indicates that if the tongue roller is capital R and the recessive is small r, so it's a homozygous tongue roller. So that means that one parent is dominant, carrying both the alleles, and the other parent has to be recessive. Now, if I separate out the alleles, I get capital R, and small r, small r. So I did my 2 steps up here. Now I'm going to show you the problem. If you now make the Punnett square, and let's just put down on the top capital R, small r, small r. And now if I fill in each of the box, notice whenever you have one lowercase, one uppercase, you always put down the uppercase first. Now what is the combination up here? All of them seem to be dominant, so all children will be able to roll their tongue, even though one of the parent had recessive. Why? Because the other parent had also homozygous dominant characteristic. Now you will practice some genetic problems using these 5 steps on your own, and you are expected to know those on the exam. Practice some on the web study or some of the quizzes that I've given you, and trying practicing with these. So this concludes our this 3rd series, and we have one shot lecture after this on genetics.